The Mango tag gel imaging methodology demonstrated here is robust and is anticipated to be able to be simply extended in terms of sensitivity and specificity.
This would further simplify the routine purification of biologically important RNAs and RNA complexes by the simple expedient of adding a Mango tag to the RNA of interest.
RNAs are being implicated in more and more diseases. Therefore, this post-staining method is an easy and fast way to be able to track and visualize RNA.
Extra care will be required in transferring the gel from one place to another, as the gels are fragile and prone to breaking.
To prepare a 30 ml denaturing gel, first select the polyacrylamide percentage with the appropriate bromophenol blue and xylene cyanol dyes compared to an RNA of interest to ensure high band separation. Then, mix solutions A, B, and C according to Table 1 and add APS and TEMID. Immediately pour the gel solution into an appropriate gel casting apparatus. Insert the desired comb and leave the gel to polymerize for approximately 30 minutes. Make up the gel tank using 1x TBE solution and carefully remove the comb. Carefully lift up the gel and mount onto the gel apparatus and secure using binder clips. Using a syringe, aspirate out the wells immediately prior to sample loading.